you so much. And so with all your a priori permission, we are recording the talk, and it will be put up in the YouTube page of Genome India International. We will take another consent before we upload it. So before we start this webinar series, may I quickly introduce Manish Pandey, who is going to introduce our guest speaker, Ilga Porth. Manish needs no mention. He is a senior principal scientist at ICRISAT based in Hyderabad and vice president of Genome India International. He has been with GII over the last 2 decades, ever since inception, and he's a pioneer of various breeding technologies in lieu of various qualitative and as well as quantitative traits. So today is going to introduce our guest speaker, Ilga Porth. Yeah, over to you, Manish. Thank you.
Thank you, Pras. Thanks for your nice words. And Ilga, so it's nice seeing you after January that we had your presentation in our PAC climate change conference in San Diego. So welcome to this webinar series that is under the Genome India International, and about this society. So it's committed towards like genomics interventions towards the betterment of crops and humans and animals. So and of course lately even environmental sciences. So we welcome you, and then to the audience here today for Ilga Porth. So let me mention that she is associate professor in Laval University, and she has been focusing on her research on tree genomics and genetics, and then of course their improvement. And also she has like say expertise or extended her work towards looking after for Canada, like how this is economical in a way that how the economical potential can be harnessed from forest genomics in Canada. So that is has been there, and so today we have opportunity to learn from her a lot and listen to her. So welcome, Ilga, again, and you have now maybe 45 minutes of presentation, and then maybe we can have 15 minutes of question and answers at the end. So thank you and welcome, and now floor is yours.
Okay, thank you very much for the introduction. I'm gonna share my screen. Okay. Okay.
Yeah.
So everything's fine. Can you hear me? Sounds good. Everything is okay. Good.
Yes, yes, very clear. We can see slides and hear you, Ilga. Please
nice. Go. Okay, I will start. So thank you very much. So first of all, for the kind invitation to give a webinar here in the context of Genome India International today. And I would also particularly thank the organizers who contacted me directly. So Professor Chitra Cole and also Dr. Yasoda for organizing this with me. Yes, as I said, I'm an associate professor at Laval University, which is in the eastern province of Canada in Quebec City Station. I'm also a member of the Centre d'études de la forêt, so the forest research center in Quebec, which is a conglomerate of different institutions that do research on forestry, actually, mostly ecologists, actually. And I'm also a member of the North American Forest Genetics Society and also of the Canadian Forest Genetics Society. And I'm also deputy coordinator of Division 2 at IUPRO. So my main research interest is actually to look at forest genomics. So understand basically the the genetic background or underpinnings of certain traits that are interesting to us in terms of economy. So fast growth, good quality, like for example on poplar, I did substantial work on poplar genomics, but also looking at some of the threats that is related to increasing threats from climate change. Which is dieback of forests? What is the genetic background, basically, of the susceptibility of trees? Can we find resilience, more resilience, plant more resilient trees? Also look at the background, what drives actually forest pests local forest pests to expand their ranges, and be also a threat to other species, not to the primary host, but also perform host jumps, because these things get more and more costly, especially in Canada, which has a vast forest, and the boreal forest is especially affected. Then we have also the problem, of course, of introduced pests and pathogens. For example, here's an example of the costs for replacement of ash trees due to the threat of the emerald ash borer. And this is mostly the problem in prairie provinces, but actually also in urban contexts. So urban trees are also threatened by these invasions. And then generally we found in the boreal forest a substantial retreat projected for conifers. So there are certain species like the black spruce, which is more susceptible to these climate changes. And they are really prognostic very negative growth outcomes for black spruce under the regular scenario that we don't do anything about more greenhouse gas emissions. So this is the most severe scenario, RCP 8.5. And then this is basically still a good growth outcome, and it gets really into the negatives here when we look more in the south, where actually the current distribution of that species is. So in general, for us in Canada, what is really necessary is to look at more resiliency in forestry species, to diversify the Canadian landscape, because most of the planted trees here are actually conifers, and the conifers are more and more susceptible to these climate changes, especially on drought, I have briefly showed before. And then we would also do more for sustainability, to really provide a sustainable wood fiber supply, not only in terms of resiliency, but also in terms of forest certification, and then timber tracking, which is important actually for us to stay in the market and to really ensure that the trees or the wood that we take out of the forest is from sustainable practice. So this is really important. And of course, the problems that we have with diseases or pests that are on Canada's doorstep. So one of the things that will be working on very shortly is to tackle the oak wilt disease, which was for years really close to the Canadian frontier in the south. So it's really devastating in the United States, but it has now, I think 2 or 3 weeks ago, really entered the Canadian jurisdiction. So I have been involved in Genome Canada projects. So these are projects that are financed federally, but then also locally by the Genome Center. So Genome Quebec, for example, or Genome British Columbia. And these projects actually aim to use genomics in forestry. So be it on trees or be it also on anything else that's related to forestry, that could also be tackling this disease or pest problem. And then actually provide, based on the outcomes, short to midterm benefits. And this in collaboration with the end users, with different stakeholders from the different federal organizations like the Canadian Forest Service or the Canadian Food Inspection Agency that is related to also plant health. So this kind of projects we have ongoing since long term, so more than 20 years, actually, Genome Canada or also the provincial genome centers are investing in this type of project. So the first start of this project was actually to develop the genetic resources first, like genome sequencing, ESTs, SNP arrays, for example, to genotype trees, and this kind of things. But we are now entering really the post-genomics area, or we have already entered it, but we can really apply this more widely, and this is also required actually in those type of projects, so that we can really see an outcome in practice. Like one example could be to increase the genetic gains. So there are several projects that use now genomic selection actually to decrease substantially the breeding cycle for trees, because as you know, trees have a long generation cycle, poplars less, but still, but then conifers really long, like 60, 70 years, also turn around. So this is something that was done and also implemented. And then we can also have the long-term benefits. And I want to mention here environmental benefits. So environmental benefits can be really, really long-term, or they are long-term. So to have more climate-adapted forests, to manage with tools to keep disease out of the jurisdiction, and also to have climate resilient and pest resilient urban forests that improve the health actually of the population who is inhabiting cities. And then we also need to look at now towards replacement species that can actually replace those ones which are already decimated, and we cannot replace them anymore actually with genotypes that are resilient from the same species. So we have to look into different species, but of course those species again have to be resilient against threats of pests and pathogens. So that's a really, really difficult thing where we are now. And then of course there is the issue of agroforestry using fast growing trees like poplars, for example, agroforestry, or using other trees that long-term can capture more carbon, like in the prairie provinces. We're also working on oak species. So coming back to my own research now, so I have these two pillars basically that my team is working on. So which is tree improvement, understanding the basis of certain traits that are important, like fast growth, but also wood quality for bioenergy. And this I use mostly poplars, but also forest health monitoring. So I'm also working on keystone species like aspen, so which is also belonging to populus. So and also oaks actually recently, because they are found to be more resilient to the changing climate. And then the other thing, my team is also looking at integrating genomics in developing biosurveillance tools for forest invasive alien species. And we had been working with 4 top priority species in a Genome Canada project. So my team has been working with the Asian spongy moth, Limantria dispa subspecies, the European one, which has been introduced 150 years ago from Europe actually into Eastern Canada and is now established actually there. And then also the Asian subvariant of the Asian spongy moth, which is not established, but it has been intercepted several times. And the other thing is that I'm working on is the Asian longhorn beetle, which is really a huge threat, and I will be talking about that. So with regards to adaptive diversity in forests, so we need to know basically the genetic diversity first, and then look which of this diversity or which of this genetic variation is actually adaptive in the forest. So to do this, we are actually looking at the genomic. Sorry, I have to move this one. We are looking at the genomic variation that is associated with adaptation. And for this, I'm using model species like poplars, like Populus trichocarpa, which is the black cottonwood, which is growing on the Pacific Northwest. I will show this later. But then also wider distributed species also longitudinally. Yes, that reaches up north from Alaska, basically down to central Mexico. And then also in terms of East and Western North America. And then also what I'm interested in is to actually look more on the different signatures in the genome. So not only SNPs, which are usually detected when we look at local adaptations, so trees or individuals that are populations rather that are locally adapted to climate. And this, of course, is also a question of the historical demography that we also need to understand. But this all takes really long term to evolve. But there are other features in the genome which evolve more short term. If we, for example, look at bigger stretches like structural variation in the genome, which can actually support the genomic plasticity, so that the phenotype is a genotype, the same genotype can actually, based on gene expression changes, can have different expression, phenotypic expression under different environmental conditions, which can actually support the adaptive capacity of a species. And then, of course, we know that structural variations are involved in hybrid vigor. So for poplars are planted, actually hybrid poplars are planted for the hybrid vigor. So they're really crops. So this is my connection to crops as well, because poplars are considered more like a tree crop, or can be considered like a tree crop, similar to eucalyptus, for example, but also certain conifers like radiata pine. And this structural variation, so this term of, because it's related to genomic plasticity, and it's also connected with the occurrence of the transposable elements in a genome, this can evolve much faster than this signatures of local adaptation. So we published in this respect too. And then we also look at ploidy changes. So aspen, North American aspen, is a very interesting pre-species to look at because it is a facultative polyploid. It can be diploid, but it can also express polyploidy, mostly triploidy, under certain circumstances. And I will show you then some results. But this is related more also to the environmental condition. We found that it's actually associated mostly with environmental conditions, especially with harsher in terms of higher temperature, but also less precipitation, so droughted environment. So ploidy can also be related to genomic plasticity based on gene expression changes. And this also takes a shorter term to evolve because you can also induce ploidy, and you can get polyploids even by doing hybrid crosses, the same as what I mentioned for hybrid vigor. So this takes shorter term to evolve than this local adaptation of SNPs. So we have been working on having the improvement of forest tree crops. So we use the black cottonwood, which I mentioned before, which is growing here in the Pacific Northwest. This is in gray, the natural distribution of the species. So there is a nice germplasm collection. In the United States, in Canada, we had several common gardens where we did extensive phenotyping on ecophysiology, phenology traits, growth traits, but also wood quality traits. My part was mostly involved on wood quality traits at the time. And then one of my PhD students, he has also looked at whole genome sequencing here from a wider collection that I have ever been able to use. He had more than 1,000 genomes actually at his disposition to do high quality SNP calling. He used two different types of SNP calling softwares like GII and Platypus, and he compared also with what has been done previously from different teams, and he did this comparison. So in the end, he had more than 8.5 million SNPs at his disposition to analyze. So we were looking actually at from a different perspective at those SNP collections, because we wanted to investigate loss of function mutations that are naturally occurring, so that they are viable. This was interesting to us. And my student found more than 8,000 stop-gain variants in over 6,000 populus trichocarpa genes. So what we actually did in here, we went back to the wild species, and then we had a reference genome at our disposition, which was published already in 2006, and it's available at Phytosome. There's already the fourth version available, 4.1. There is an extensive gene expression atlas too, and we had also other gene expression information available to us. We had genotyping data by SNP calling, but we had also previous genotypes, and we had extensive phenotypic data. And then what we did with all this, we looked into these candidate genome regions. And our allele mining, as I mentioned, was really targeted towards stop-gain variants. And for this, you really need to have high-quality SNP calling. And this is what we did. Some of them were rare, some of them were not. For us, this was also interesting because we connected this with some of the wood quality traits, and we found some nice association especially with hemicellulose. And hemicellulose is something that is basically also the glue together with lignin actually for the secondary cell wall, but it is a problem when you would like to extract the sugars that are actually, especially the glucose sugars that are stored in the cellulose. So to have individuals with less hemicellulose is actually favorable, and also the structure they have less decoration with certain residues is also something that is favorable. So we found some association, and this association was actually explaining more than 10 or approximately 10% of the phenotypic variation, which is quite good. Another thing that we were looking at was the adaptation in populus trichocarpa, and for those we actually used the SNP chip that was now 7 years ago, 8 years ago. We tested around 30,000 SNPs, and we found around 3,000 SNPs that were under selection. But what was really interesting to us here was to see actually how many of those SNPs that were under selection when we did the selection scan were also candidate genes under the GIVAs, which means genes that were associated with certain traits, but those traits needed to be also adapted. And to figure out if those traits like biomass, ecophysiology, phenology, pathogen resistance, or wood traits are really adaptive. So we did this testing for non-neutral QST, which is comparable to an FSD, which is a fixation index between the populations. So basically, the population have to be differentially in terms of their adaptive traits based on this non-neutral QST. And if they are, then we can say they are adaptive traits. And those QST values should be also well above the neutral FST values. So what we found in the end, we found 118 SNPs associated with 81 genes that were outliers under the diversifying selection, but they were also associated with the adaptive traits. In Aspen, we did different things. So first of all, we had a huge collection of individuals that ranged all the way basically from all the distributions, with a bit of holes also in the collection, but still. We were the first actually to figure out that there are 4 different genetic lineages in Aspen. And we call them Northeast North America, Northwest North America, Western US, and Mexican clusters. And you can see here the differences in the fixation. So let's say these 2 clusters here in the East and in the West are less differentiated than all others. And by comparison in Mexico, you see huge differences between the genetic lineages. In addition, once we figured out the overall genetic structure of the species, we also looked within those 4 clusters at genes or SNPs under selection. And according to the cluster or related to the cluster, we found different numbers of SNPs that were outliers, so to say, between 25 and 410. The least we found here, actually, interestingly. And then here is an example of a gene that where you can see the allele frequency distribution across the range of the species. And you can see the differences. So there is more variation here, and there's more homozygosity here. I have to say that the SNPs that were used here were based on GBS. GBS is a reduced representation sequencing tool. And we used about 40,000 high-quality SNPs, but we didn't use millions of SNPs. But still, we found some interesting results. This gene, the LAX1 gene, is a long-chain acylcoenzyme A synthetase, which is implicated in the formation of cutin and bags on the leaves. So this is interesting because we have been suspecting that this has really a functional role. So this association that we found in relation with adaptation might really be functionally relevant. Because I told you this was based on GBS data, we have to take the SNPs that we have been available to us based on the restriction enzymes that are used in this kind of method. So here we found a 5' UTR distribution that we here mapped. But we also looked at the expressed region of the gene. So the coding sequence from the RNA-seq data, and we could find a similar pattern. So for this region here, which is here framed in red, we found this variation. And then for the Mexican cluster here, we found way less variation. So there is really a linkage, basically. So, and we will keep looking at this further also in terms of phenotyping those two. We also found an interesting correlation between the density of the stomata that was tested in the 4 genetic clusters, and there were differences, significant differences, and those differences were actually correlated with the mean annual precipitation, meaning that you have more stomata, you have more precipitation, and you have less stomata, but there's less precipitation, and this is actually a strategy of the plant to conserve the water in drier environments. We looked also at ploidy. Ploidy was interesting to us in Aspen because, as I mentioned, this is a facultative polyploid, so it could express ploidy under certain conditions. And actually, when you look at the distribution here of the triploids in green and the polyploids here marked in blue, which is the proportion of the triploids here in the population, that here, in comparison, you have the annual heat moisture index, and dry and warmer environments have a higher index, meaning that here, following this color scheme, you have here really the drier and warmer environments. And you can see here also the distribution or the occurrence of triploids is higher. And the correlation says the same. There's a correlation basically of the ratio of triploids per population with the heat moisture index annually, and also some heat moisture index. We also did now landscape genomics by looking at the climatic variables specifically. And based on the climatic variables, we performed PCA, and we used the first 3 principal components. As you can see here, the first 2 express around 73 or 74 of the overall variation in climate. So the first PC explains 58% and the PC2 15, but we used the first 3. We used 2 different scanning tools, BIAS-ENV and LFMM2. Both are correcting for population structure. And we found here on scaffold 3 of the genome nice actually volcano pattern, so to say. So there's really like a sweep of SNPs that are associated with the climate PCAs. And I mentioned the LAX1 gene previously, and this LAX gene is also found in the scaffold 3. And the scaffold 3 has an interesting feature because we found here major genomic sequence inversion. We found also that actually Tremoloides has a higher percentage of inversions in terms of structural variation than other porphors so far. And this is interesting. This seems to be a specific feature of that species, I would say. And also because there is like a block of this adaptive variation, we can see there is really repressed recombination for locally adaptive SNP. We also wanted to understand the phylogeography of the species to better understand basically how resilient the species can be from demographic history and what is actually the diversity in the species in terms of the effective population size or the current population size in comparison with the ancestral population size. So we actually tested 4 different scenarios within each of the 4 genetic clusters using fast simple. So we tested stable population size, or with the occurrence of bottleneck, population expansion or contraction, comparing the ancestral population size with the current population size. So we excluded stable population size and any contraction of populations for the 4 genetic lineages. But we found for the Northeast, North American cluster and the Western cluster and the Northwest North American cluster, expansion of population compared with the ancestral population. But for the Mexican population, which is really growing up high in high altitude, like over 2,500, 3,500 meters, population, a bottleneck actually, population bottleneck. And then after that, an expansion of the population again. We also tested 16 different scenarios between all of these 4 clusters using do-it-yourself ABC random forest, because we wanted to see actually more to the demographic history so the split basically between the different lineages. Out of the 16 scenarios, actually the most simple scenario 3, which didn't include any ghost population, any ghost refugee populations, that remained actually the most significant one. So we can see that the first split from the common ancestor occurred from Mexico. Then the split in Western US, further to the left, as I had showed you. After that, this population split away from the Western US population. And then finally, the Northwestern population, which is here up there to the west, is the most recent population. So we looked actually in Populus balsamifera, more specifically at structural variation, and we tested 2 different crosses. They are polymix crosses, meaning we have here crosses from within the population way up north in Quebec, really high latitude, and then comparable inter-provenance crosses across, also with a polymix, where the female parent is again from here, and the polymix, so the pollen comes from East Saskatchewan. So we expected to see a huge segregation of adaptive traits here because of the geographical differences, so the climate differences. And this is what we found here. By testing around 19,000 genes in Populus balsamifera, which is widely distributed across Canada. We found over 1,700 genes in copy number variation, and we found also 22 hotspot regions. And then we had at our disposition several traits that are related to phenology, like bud flush, green tip, leaf emergence timing, bud set timing, but also ecophysiology traits. And amongst those ones, we found bud flush, green tip, leaf emergence timing, bud set timing associated with genes in copy number variation, which you can see in the circles plot now plotted from the outside to the inside. The two ecophysiology, photosynthetic rate and intrinsic water use efficiency, you find here at the two most inner circles of that plot. With regards to Populus tremolidus, the species I talked just before, we also do whole genome sequencing. We just finished the reference genome sequence, and we have also been looking at copy number variation using Oxford Nanopore long-read technology. And here in the circle plot, we see here the gene density across the genome and the 4 individuals from the 4 different clusters that we use in terms of long-read sequencing. And you can see here the read coverage. And comparing gene density with read coverage, you can now also look at genic, so structural variation, so like genic copy number variation. So one of the things that seems to be actually relevant for poplars in general, because other groups also looked at structural variation, is that deletions were most prevalent in this family, basically, of species, in this genus. And but here for this particular species S, we found that the insertions were relatively high. So they made up on average 40% of the proportions of structural variation, which is really high because they are much lower, usually 15% in other poplar species, or in some species they found even none. We have also performed stress physiology experiment by looking at trees. So we had over 100 trees in our experiments where we looked at gas exchange. We did gas exchange measurements, and we found some interesting differences between the individuals from the different genetic clusters. And we did also RNA-seq experiment where we saw differences between the 3 clusters that we tested. So we didn't test one cluster because the trees were not at the same age, and also this cluster was very similar to the one in the east. So I showed you before on the fixation index that those were not very differentiated. So we didn't take the northwestern cluster to test. So we saw differences in gene expression between the different clusters and also in response to temperature and water stress. Another things that I would like to briefly touch on today is experiments that we did that could be useful for breeding, that could be useful also for chemotyping, so using basically metabolomics or specific metabolite profiles to check for susceptibility actually in species to invasive pests. We have done this with the Asian spongy moss. The Asian spongy moss is highly invasive. It is a Dirus defoliator for oaks, which are one of the most susceptible species of this of the species complex, actually, because there are several sublineages as well. So there is the Asian sublineage here, which is flight capable. It also has a bit of a higher host breadth. And then we have the so-called European lineage, which has been introduced, as I mentioned before, in Eastern North America and is now established. So we have been doing a pilot where we were interested to compare the European lineage, which was a culture basically that was maintained at the USDA, a U.S. Forest Service containment facility in Connecticut, with another strain from Mineralny, which is here in Russia somewhere, which is from the Asian strain, so-called Asian subspecies. And we compared two hosts, the oak, which is the preferred host, against Pinus strobus, which is a conifer species, and we found differences for the Asian strain in terms of host preference, so what we expected, but we didn't find anything for the European species. And for us, the Asian species, because of our project and the species that we selected to investigate, we continued to work with the with the Asian strain. So we found here differences in the development of the insect larvae, which was feeding on the foliage in terms of body length, body width, and also head width on the Asian strain. So we did a full-blown study, I would say, of the 10 most important conifer species, economically important conifer species in Canada. And then we always ran the experiment with black oak as a positive control. So what we found in terms of larval weight gain, it's day 10, that of course, as expected, the larva did the best on oak. And I have to say also, here in this experiment, we were looking at 2 different strains from China, from Beijing and also from Harbin, which is hundreds of kilometers further north, and it's a colder climate too. So to see some differences in the strains, so we see some differences also within the strains, but for us it was interesting here to figure out which of these conifer species Canada would be more susceptible, and we found Douglas fir, but also lodgepole pine here most susceptible because the larval weight gain was the highest for the conifer. And then we see Pisces, Norway spruce, the least favorable, and we can also explain this here because the district. So Norway spruce is a plantation species, important one here in Eastern Canada, but it's an exotic species. It grows here actually in Europe, in the Alps, and then also here in Scandinavia, and here Russia too, and then it hybridizes with Siberian spruce. But basically, we think there has been a co-evolution basically between the insect, which is from there, so from Asia basically, and then with this conifer species. This is our explanation. And we found one particular terpenoid that we tested in the foliage highest across all those conifers. It ranked highest, and we found it really high, especially in the 3 spruce species, among them also Norway spruce. So we are able to separate actually those conifer species also in terms of their profiling, in terms of metabolism. So here we focused on terpenoids. We also did actually phenolics, but here I'm presenting terpenoids. We did also an RNA-seq experiment on the moth or on the larvae while they were feeding, and we found a nice separation here in terms of the 2 strains. This is for Harbin and this is for Beijing, but we found also some separation on the different hosts they are feeding, notably on oak and some other conifers that were separated. And for boron acetate, we found really those differences in the content of the different species, host species actually. And then we were able to associate the occurrence of boron acetate in terms of concentration with the expression of the number of significant genes in the midgut of the larvae that were feeding on this foliage, which is interesting for us. And here you can see as an example one gene where actually the expression of this gene increases with the amount of boron acetate that is found in the species. So there's something going on, and we are still working on the finalizing of the analysis. So these were horticultural plants, but we have also been looking at the adult trees, and the adult trees actually need to show us what is really there in terms of defense mechanism, defense compounds in the breeding program. So we see that there is a difference in the horticultural plants that we used for feeding, for example, Douglas fir, and then in the breeding program. So we found terpinolene, which is this one, and then sabinane are actually in much higher concentration in the breeding, which is interesting. And then we also were able to use this breeding program individuals. We had 17 families to estimate the genetic correlations. Here you see the estimation of, or the network of the genetic correlations of the monoterpenes actually. So we found here negative correlation, but also positive correlations. And depending on the importance of the compounds, but also in terms of the heritability, we can make inferences or give advice to the tree breeders actually, and the importance of those compounds for breeding. So lastly, I will briefly touch on something that we're also working on, which is using genomics, integrating genomics for biosurveillance tools. So one of the things, and this was especially done for the Asian longhorn beetle where I was involved. So the interest is here from a management point of view, but also from a research point of view. So what is it? So what is the intercepted individual that we get to analyze? We need to know the species, because there are some species which look similar. So for example, the citron longhorn beetle and the Asian longhorn beetle look pretty similar. They have been suspected hybrids, but we recently found out that the species can actually not hybridize at all. These are not hybrids, these were just local or ecotypes actually of the Asian longhorn beetle. Then also we were interesting, where is the source from? So where is the individual from? So we can do population genetics and population clustering to figure out where they map on the distribution of the native range of the species of this pest, for example. And then also we can look at the pathway and to understand how the spread was done. So we can identify something like bridgehead populations, where these populations can be the source of another invasion, for example, and so on. And then we are also interested to see at some of the traits of invasiveness. I have shown you this before, like something that is related to host utilization, but also climate adaptation in terms of code adaptation. So here you see the distribution of the sampling of the Asian longhorn beetle in China, and we also sampled in Korea. And this is all the listing of the invasiveness of this species in North America, but also in Europe. So we have done our homework in doing the basic population genetics of the species in the native range. In China, we found a north-south gradient, but we also found distinct 6 clusters that we could now use basically to define regions and use the markers as well to be able to assign, based on those markers, unknown individuals. So our testing of the accuracy of assignment to the north-south region here necessitated only basically a handful of markers. This is the number of markers that you actually need to achieve over, this is the baseline of 90% accuracy of prediction to reach this. For those regions here, you need already more markers, like at least 100. If you want to go beyond 90%, you need around 200 markers, and actually we use 300 in our biosurveillance tool. You can also do again scanning for adaptation and see if you have interesting candidate genes, like in our case, glycerol kinase gene, which is actually important for the usage of glycerol, which is a osmoprotectant towards cold resistance in the beetle. And you can see really this missense mutation is really gaining in relevance here in terms of allele frequency the colder it gets here. So this is interesting. And then, of course, we want to assign now and see where this incurred intercepted individuals come from. We have here sampling from North America, from Toronto, Canada, but also from the northeastern United States. And we were able actually to see where the most of the invasion came from. They came from this region, North 2, as you can see here in this assignment plot, but also some from North 1, which is here, the Beijing region, and then one from Massachusetts. We can find also incursion from the south. So you can use these genomic markers. So I want to stop here because I think I've been long, and I want to draw some conclusion briefly. So we have used really genome sequencing from whole genome sequencing to test if it is possible to use breeding with rare defective alleles. And we saw that we have to use a lot of meticulous validation steps, including different layers, genome data, data in terms of gene expression, where we found there is differences in different environments with gene expression, and also association with phenotypic traits. And if this association hold true for different environments. Then I show that we can use chemotyping to inform current tree breeding programs for species at risk. And also we can extend the approaches now, expertise that we develop into this biosurveillance approach for forest invasive alien species, to also use this approach with assignments, also to apply to trees actually in terms of traceability tracking. So to assign basically wood samples to the original populations as well. So we call this the timber tracking approach. And our Aspen Genomics project has actually expanded. We are now collaborating via the University of Berkeley with the GII to sequence 2,000 individuals. Berkeley is performing extensive greenhouse experiments, and so we can better understand the biology of these keystone species for future preservation and management. So I want to thank you, and I also want to place those acknowledgements of the people working on Poplar and my collaborators, the funding agencies, the forest health research that has been led by 2 students and collaborators, and so one of my team members or 4 team members. So here I close, and thank you very much.
Yeah, it was very, very good talk, and then informative also, because many of us actually working on mostly crop genomics and breeding. So when you bring perspective from forest genomics, and also you were showing like some different surveillance also, like how you track in those areas. So I'm not sure. So all the audience, maybe you can put your questions if something is there, so I can ask on your behalf. Meanwhile, Ilga, I have few questions to you. Maybe you'd like to answer. So the first one is, like, when you go for reference genomes and you are doing genotyping, or when you are sequencing and then analyzing, so what kind of problems you see in forest genomics because of heterogeneity or heterozygosity, or what kind of the problems are deployed even?
Yeah. So there is actually, because we were actually doing our whole genome sequencing with an institute who is focused on crops, and they were not really used to such heterozygous individuals or species like aspen or poplar. So they ran into several problems, and but they told me it is really a nice experience for them. We had to use 2 different types. So actually, in the end, we ended up with doing 3 types of sequencing. So of course, we actually 4. So we had to do we started with ONT, so with Oxford Nanopore, but this was not sufficient actually to properly resolve our contiguous genome sequence. And then, of course, we did high-C to do the chromosome assignment, and then, of course, the polishing with Illumina paired-end sequencing. So we had to do PacBio 2, and we had to do very recent, and we just got back a couple of days ago the latest assembly. For Poplar especially, and then there is another thing is that in Poplar we have this whole genome duplication, so we have a lot of duplicated genes as well. That is problematic to resolve as well. Yeah, so this is so for us, because we have a collaborator who is very much interested in chromosome 19 on Poplar, which is the sex determination region. Because Poplar is a dioecious species, that means there is female and male flowers on different plants. And he's looking at the evolution of this region, mostly all across poplars, but starting with aspen. And they are different between the different aspen species too. So there are some duplications of a certain gene which couldn't be resolved with our first assembly.
Yeah.
So it is it is complicated depending what the focus is. So we went into the more high price, high cost assembly that we could do.
I think that's good that basically when this sequencing began for simpler genomes, I mean to say, now we have technology, high-C and others that which can help even making genomes for complex and larger genomes, and also of many other complexity that you explained. There is one question here coming. Maybe let me read here. It's from Kriti Rao, and she says, what bioinformatics tool have you used apart from NGS? I also not clear. I'm not clear.
So you mean apart from next generation sequencing? Well, actually, we to be honest, well, I used actually when I did postdoc, and I used actually SNP chips from Agilent. That was the first Poplar. That was the adaptation I showed in Populus. So we used basically candidate genes that were focusing on certain pathways in wood formation, a bit of adaptation too. So that's not next generation sequencing. However, to select those SNPs, so the SNP ratio, we had to do RNA-seq. So this is again next generation sequencing.
Okay.
Well, we did, which is actually the part that I showed you with the structural variation in Populus, Populus balsamifera, so the balsam poplar with a wide distribution, and the circus plot, which is we used again an array basically based on probing. But then we had to use the whole genome sequence, Populus trichocarpa, as the reference to select basically probes that cover first most of the genetic regions. And then we had 1 million probes, and then we selected the best ones and ended up with 19,000 genes that we were able to. And they were covered on average with 8 probes, those gene regions for our structural variation. Yeah, but for the rest, even genotyping by sequencing, right, we use either ion torrent or we use Illumina for sequencing. So we use this mostly.
Thanks, Ilga. And we have 2 questions from Pras. Maybe Pras, can you ask yourself? So in place of reading it, that would be fine.
Yeah. Thank you so much, Manish. Thank you so much for an excellent talk, Ilga. I was just wondering, when you check the insect adaptation rates, would you also be interested to look into the horizontal gene transfer events that are emanating? I mean, so for instance, would you be interested to check any genes that might have evented lateral gene transfer or horizontal gene transfer between the insects and the plants? Yeah.
Well, this looks interesting. We haven't looked at this actually. But what we did is when we did the RNA-seq experiment with the Asian spongy, mostly Mantria, we found indication for some virus, and depending on the individual from that colony, because these are actually colonies that were maintained over lab colonies, right? That were maintained over generations. We found actually evidence of viruses that were there, but we didn't find any negative outcome for. Well, if we found more virus in one, we didn't find a negative outcome in the population, but and also our collaborator in the US, so the population was healthy still, but we found indication of viruses there.
Excellent. I have another question, something on these lines. We are, in fact, I'm a human biologist, but an opportunist plant biologist, because I keep collaborating with some of the plant scientists as well. But since I work on human cancers and human rare diseases, we are largely interested in looking into the genetic variation, that is sitting in the non-coding regions of the human genome. So if you really take an analogy, there could be certain genetic variation, attributing to non-coding regions. So there could be a mutation, that might be, say, sitting in certain non-coding chunks. And let's assume that mutation harboring gene might be, say, overexpressed,
Hello, I think Crash is frozen.
Yeah, maybe he has some internet issues. Yeah, he's back.
Okay.
Yeah, would you also look into such kind of scenarios? I mean, where there could be a gene, that might be overexpressed, and yet, that's because a SNP in it might be, mutated, or there is a SNP, in it. Yeah.
So are you referring to plants or?
Yeah, in plants. Yeah.
So if that is mutated, yeah, so that would be something related to a cis effect, right? So so we looked at this actually in Populus trichocarpa. We looked at that. So we looked at the mutation, we looked at the expression changes actually if there are changes in expression for individuals that house the mutation and those ones which don't. And then we also looked at allele-specific expression, because most of these mutations were actually heterozygous. So we could look into allele-specific expression. And we didn't find that this adverse, let's say, disruptive mutation was in any way suppressed. And in any way, the expression levels were relatively comparable to so it didn't change anything in the overall gene expression. That was interesting to often. But what we did find in the environment, we found differences even in the same phenotype, because we could have clones, and in these plants we can have clones, the same phenotype that was propagated clonally, so the same, right? And then planted in different environments, and even there we found differences in the expression, which we found interesting in terms of allele-specific expression. So there is, and this regulation we really don't understand yet at all, actually. Yeah, but that's a good point that you raised. Yeah.
Right. Yeah. Thank you so much, Ilga, and thank you, Manish. Yeah.
Professor, I have few clarifications. Like adaptation variations. We do work on tropical trees like teak as well as sandalwood for the adaptive variations. We are scanning the population throughout the country, and we are looking at the variation through whole genome sequencing. Here I have few clarifications, like we could identify the diversity hotspots. And here most of the forest areas are with the forest departments. That is, the forest managers only handle this. Being a researcher, this information of genetic diversity hotspots I need to convey to them. And in the scenario of climate change, how do they take these results and go ahead? Can you throw some idea from your experience?
Yeah, so this is a great point. So I would say this work that we're doing is the first step. So we are now doing in Aspen, so that big we actually are now working on relating this to climate models. To see, we use those SNPs that we identified that are adaptive now in models to actually predict which of the populations in the future with future climate models in Canada, or actually the distribution range of the species, are maladapted. So this is called like genetic or genomic offset. So this is what we are just now actually working on. We haven't finished it. I would have loved to present it. So this is actually our next step. How we can use actually those SNPs under selection, these adaptation hotspots, right, that I showed in Aspen, like the scaffold 3, for example, to see actually to predict basically the adaptive capacity into the future. So we are actually working on this right now as we speak. But this is the next step actually to so we use basically, I don't know if you heard about it, but there are many others or several others. We use gradient forest actually to do that.
Right. Then one more query, like you were mentioning about timber tracking.
Yes.
Like for example, again, teak, red sanders, there are two different species. So they are highly valued timber. And again, the forest managers are interested in timber tracking. We are trying to develop SSRs as well as SNP markers. And handling SNPs will be a bit difficult for the legal purposes, we feel. So can we use the SSR markers itself for timber tracking? What is your opinion?
We haven't used this. The reason is too well. Yeah, so because of the problem with this NALA lease in SSR, that was to our stakeholders or the Canadian Forest Service, that was a problem.
Okay.
And for SNPs, we see more flexibility. There is something called mass array, where you can actually multiplex and use a manageable amount of SNPs that are highly targeted, actually, in terms of species, population. And also you can add adaptive SNPs. I don't know how. Well, if you have, for example, so my question is, your SSRs, are they in genes like EST SSRs, so-called, or are they in non-coding regions? So because maybe EST SSRs that are in coding region could still be affiliated with adaptation, because we wanted to give further indication with our tracking tool, basically on the origin and also and the adaptability too, because we would like to use it also in a forest certification tool for Canadian forests now. So we need to know if this individual or this population has the markers, the adaptive markers that we are after too. So this is why we use SNP, and we use the mass array tool, which is a single nucleotide extension reaction, also PCR based like SSR. So you don't need a very high quality DNA actually. So it would be useful if you extract the DNA from wood itself, right? You're extracting the DNA from the wood eventually to use it.
Right. We have the SSRs from both coding as well as non-coding regions. I think we should be able to go ahead with that. Thank you.
Yeah.
So Elga, maybe we are late by 9 minutes, but one small question, maybe if you want to take that, and then we can close after that. And the question is that one of the audience says that we have done our work in population dynamics in fishes in our locality. Kindly suggest what kind of bioinformatics analysis to be used.
So in terms of population dynamics, yeah, I think everything that, well, you want to look at the effective population, you want to look at the diversity, right? At the diversity, you want to see if it is an inbred population or not. And so usually all this, I don't know what type of genomic data you have, but I think Stacks has a lot of tools actually that can give you some first insights into those population measures and this genetic measures. And then yeah, there are lots of tools out there. If you talk about population dynamics, so this I would say is in terms of the evolution basically of the diversity. So in terms of the effective population side, this is what usually conservation geneticists are looking at.
Thanks. What we will do, maybe we can put your mail ID while putting in YouTube in our website.
Sure.
And maybe not only the friends right now present in this webinar, maybe when other friends will be seeing in YouTube, maybe you will have many more questions. They can approach directly to you.
Yeah,
And
no
also
problem.
it's just not only like sharing and exchanging the knowledge and expertise, but it's also like if some collaboration can be established also, that we also good. That is also a benefit for such kind of interactions and webinar. So maybe thank you, Ilga. So we would like to thank you from GII and also on behalf of our all audience that are present now, and that those audience those will be looking again, seeing these videos and having interaction with you. So thanks from all, and we'll look forward to see you in some other platform and have interaction with you.
Yeah, thank you very much for the invitation. Have a nice day.
Thank you.
Thank you, Dr. Ilga.
Thank you.
Thank you very much. Yeah. Thank you, Manish,
Thank
for
you.
being the host. Thank you so much. Yeah.
Thank you, Pras. Thank you.
Bye.